Hi everyone, we are halfway through the term now. We're starting week 5, so now we're on the downhill slope. Well, what did we do last week? We looked at cellular respiration, and cellular respiration is the process that all aerobes do in order to make ATP molecules. So aerobes, welcome to my neighborhood. The dogs that you hear chirping in the background are my neighbors, and they're always outside barking. Anyway, and I wanted the window open. So cellular respiration is a process where you take your food molecules, for example, glucose, break them down in the presence of oxygen, that's what's so important, and you make ATP or adenosine triphosphate molecules. Now, what's the importance of these molecules? They're used by enzymes. The energy is used by enzymes to make and break chemical bonds. And enzymes do all the biochemistry in cells. So cellular respiration is terribly important. It's really what you think of as your metabolism. This week's topic is DNA. DNA, my little buddy here, DNA, deoxyribonucleic acid. And what is it important for? Heredity. It determines the traits that you have as an organism. Whatever organism you are, if you're a bacterium, if you're a redwood tree, if you're a paramecium, if you're a human being, if you're a potato, all of your traits are found in your DNA. Very important, and very important these days. For disease detection, we can look in your genes now to see what disease genes you have. We can look at heredity to see who you're related to. And this is very interesting because I sent off my DNA about 2 months ago to Ancestry.com and found a relative that I didn't know that I had, which is interesting and exciting all at the same time. Forensic science, who did what? Who did the crime? Who left DNA behind at the crime scene? We use this in so many ways now. Detection of viruses. I'll talk about this when I get to PCR. Okay, so this week is DNA. You have a milestone due this week, milestone number 2. I just finished grading milestone number 1. Go back and read the rubric, read my comments. Fix your milestone 1, and this is very important for about half of you, because I made significant suggestions for significant changes in your topic. So make sure you look at this and fix this before you move on. You are going to need all the information from that milestone 1 for your final project, so you might as well fix it now. The correct format for milestone 2 is bullets or outline form. So you don't have to write everything out in sentences unless you want to. I'll read it if it's in sentences, but bullets are better. It goes boom, 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 boom. What are you going to do? What are you going to do? What are you going to do? So use the correct format. You're going to include all the steps to your experiment. So this week is experimental design. So what are the steps to your procedure? And you need to get very specific. Exactly how much salt are you going to add into cup number 1? Exactly how much salt are you going to add into cup number 2? Exactly how much water will be in cup number 1? Exactly which cities are you comparing air pollution in? Exactly which parts of the body are you going to be touching with the paper clip? And for how many seconds? And how many subjects are you going to have? You see what I'm saying? Be very specific about the steps in your experiment. You're writing out your procedure. And that will be done in week number 7 after I grade milestone 2. Okay. So for the only wet lab that you have this week, the lab where you're working with tubes and liquids and that sort of thing, this is the cheek cell DNA extraction lab. And you can see my results over here on the right-hand side of the PowerPoint slide. You'll notice that DNA is sort of stringy and slimy looking. It's found in the middle of that first big tube, the 15 mil tube. And it looks sort of like a little gelatinous mountain in the middle. And then in the Eppendorf, actually that's a PCR tube on the right, my DNA is up toward the top. But you can extract DNA from your own cheek cells with this very simple experiment. This is done all the time. Think about any type of forensic science show that you've ever seen. And what do? They take that swab, get inside somebody's mouth, and perform a buccal swab, and that's to collect cheek cells. When I ran my Ancestry.com DNA, I spit into a tube. Why? Because my cheek cells went into that tube with my saliva and they extracted DNA from those cells. So this is a really cool experiment to show you how easily this can be done. Sometimes in this lab, they go back and forth between cc's and mLs, and I want you to realize that they mean the same thing. cc is a cubic centimeter. A cubic centimeter is the same thing as a milliliter. So they are interchangeable volumes, just so you know that. Another hint for this lab is that your ethanol has to be extremely cold. And I don't mean your refrigerator cold. I mean freezer cold. So at least put your ethanol into your freezer overnight. A couple of days is even better to make sure it is cold chilled all the way through. And bring it out right when you need it. Don't let it sit out on your counter for an hour while you're doing this experiment, though it won't take you that long. But don't let it sit out. It needs to be ice cold because then it will precipitate out the DNA from the aqueous solution. If it is room temperature, you can add it until your tube is spilling over with ethanol and your DNA will not come out of the solution. So it has to be extremely cold. This is a very quick lab. Thumbs up for that, which is really good. But of course, you've got your milestone 2 that you're going to work on and a PCR simulation. Sorry. And you're going to answer the questions for that in the PCR lab worksheet. Lab number 5 also has its own lab report. Okay? So let's take a look at PCR for a minute. PCR stands for polymerase chain reaction. Chain reaction you can get, right? One thing happens after the other happens after the other. You get that. But what does polymerase mean? Poly means many, mer means subunit, ASE means enzyme. So this has to do with an enzyme that puts many subunits together and does what? Makes DNA. So PCR is a technique where you make lots and lots and lots and lots of copies of DNA. There are 3 basic steps to this process. So this is a technique that is used to copy DNA. And here are the 3 heating or cooling steps. The denaturation step is where you break apart, here, go back to my molecule of DNA, the long strands of the DNA. Okay, you're breaking them apart right here in between these bonds. In between, one minute, right there. In between the bonds right here of the bases. So that you have 2 long strands. And that way you can get to those inside bases. The annealing step is where you attach primers to either side of the target DNA area. So this is important. You don't copy all of the DNA. That's not the point of this. You copy certain areas of DNA that you're interested in. So in forensic science, you're going to copy areas of DNA that have a very high level of variability. So my DNA in this section will be different from your DNA in this section. So that way we can tell the difference between us. It might be that you're looking for a disease gene. So is the sickle cell anemia gene in my DNA? You can look at that specific area of DNA. Are you looking for viral DNA? That's a possibility. Is it there? So primers are usually pieces of RNA. And they're tiny little pieces. They're just a few bases long. And they stick to the beginning and the end of the DNA target area on the 2 different, here you go, different strands. One on one strand on one end and one on the other strand on the other end. These are the primers. They attach during the annealing step. And then you have the amplification step. Well, why in the world do you have to have primers? Because they get the process started. The enzyme that you use to make the new DNA is polymerase. And polymerase has to have an existing piece of genetic information to get started with the copying. And that's why you need the primers. Have you ever primed your lawnmower before you cut the grass? You know how you have that button and you push it and it pushes a little gas into your engine and then boom, it can get started like that? Well, that's why these are called primers, because boom, they get the process started. So the third part of this reaction is amplification, where the DNA, that target DNA sequence, is copied and you make new DNA. Hello, doggy. This has been in the news quite a bit lately because reverse transcriptase PCR is what is being used as one of the COVID tests. So this is the test where they stick that swab up your nose, which I've had done a couple of times already now because I'm part of one of the vaccine trials. So I've either gotten a vaccine or a placebo, both my shots now, so we'll see how that works out, but I've had this test. So what is the swab doing? The swab is picking up cells from inside your nasal cavity, and they want to know whether there are viruses present in those cells. Well, this virus contains RNA, and we haven't said anything about RNA in this whole process. Reverse transcriptase is where you go from RNA to DNA. Okay, so if there's any viral RNA there, then the first step in this process is to make viral DNA, and then you can run PCR. And then you copy that viral DNA over and over and over and over and over again to the point where you can actually detect it, whether it's there or not. So this is one of the tests that's used to test for COVID. You're looking for the presence of actual viruses right then. Okay, so that tells you whether you are actively infected at that point in time or not. The other test that they run is the antibody test. So it takes your immune system about 2 weeks to build up a large volume of antibodies to viruses if you're infected. So if they take your blood and they look for antibodies, they're looking to see if you've had an immune reaction to the viruses. So what does this tell you? This tells you whether you have been infected or not. It means that you have to have had time for your immune system to react. If this is at the beginning of a COVID infection, you're not going to find antibodies yet. You have to wait until the person has time, their immune system has time, to make antibodies. So the tests are used for two different purposes, if you'll think about it that way. So PCR, that's why you've heard of it a lot in the news lately. It's used for lots and lots of applications. And then finally, next week, we are going to be growing what are called fast plants from seeds, and you're going to be growing 2 sets of them. This is going to take at minimum 7 days to grow your plants, because your plants have to grow for 4 days. And there's 1 day in the middle when you can count the plants of one set and start the plants of the other set. This is a minimum growth time of 7 days. If I were you, the day after Thanksgiving or that Saturday, I would start my seeds. If you wait even until Sunday, you're going to be going all the way until Saturday at a minimum for all of your plants to grow. Start this as early as you possibly can. There's not much that you have to do every day. Most of the days you just check to make sure that there's water on the filter papers in the dishes and everything's okay. But you still have to tend to these every day for at least 7 days. So get as early a start as possible. It tells you that you need to use a fluorescent light. You really don't, because we all have LED lights now, and they are much cooler than incandescent lights. So use an LED light. You know, all of us have them now. I have them in all of these 3 lamps that are around me right now. So just use an LED light over your plants, and that is cool enough where it won't burn up your plants and it won't evaporate all of your water. So just know that when it tells you to get a fluorescent light bulb, you don't have to do that. And as always, this is going to be a busy week, but so are the next 2 weeks. If you need anything, if you have any questions, if you run into any problems, please email me. I'm always checking my email, m.sigmon@snhu.edu. I am here for you. I will help you any way that I can. I'll be checking even over the holiday, even over Thanksgiving, even though I'm doing a lot of cooking. I will keep checking, okay? So let me know if you need anything during the week. All right? I hope you have a wonderful week and a very, very happy Thanksgiving.